Okay, the first of a series of bite-sized lectures on digestion and the gut. The gastrointestinal tract is often referred to as the gut. Indeed, there is a scientific journal with that name. This is what we're looking at in this particular lecture, from the mouth down to the stomach, including the stomach. Okay, so general anatomy, you can find any textbook, of course. The main functions of the gastrointestinal tract are to digest and absorb ingested nutrients and to excrete the waste products of digestion. Most nutrients are in a form that is either too complex for absorption or they are insoluble and are therefore indigestible or incapable of being digested. The process, as which we're aware, starts with mastication of food in the mouth and then transfers to the stomach. So digesting the macronutrients. Would you eat these raw? Is a rhetorical question. You recognise the large molecule at the top as being one of the two forms of starch, amylose. We can't utilise these macromolecules until they've been hydrolyzed into individual glucose molecules. Cooking starts this process, and it is continued in the digestive system until glucose molecules are released. These are typically either stored or used for energy, a process which involves each of the 6 carbon glucose molecules being hydrolyzed to 3 carbon pyruvates. And you look at this in more detail in the second year when you discuss cellular respiration. Some of the proteins. Proteins are digested by hydrolysis of their peptide bonds, ultimately leading to individual amino acids. But initially the smaller amino acid chains called peptides. First stages of digestion of simple lipids involve the breakdown of the triglyceride into various fragments and ultimately glycerol and the individual fatty acids. Storage. We don't keep a collection of proteins for use. We do have what is known as the amino acid pool, a collection of amino acids ready within cells for use in protein synthesis. When a protein is needed, it is synthesized via processes you will discuss in the cell biology module. Proteins often have short half-lives, sometimes of the order of just a few minutes, after which they are hydrolyzed back to amino acids. There's comments there on the storage of the other two groups. Okay, don't panic. Now remember this is just shown as a diagram of the key metabolic pathways, particularly those for energy metabolism. Metabolism is complicated, but we can break it down into easily digestible segments which will help us understand what's going on. Again, as I mentioned, you look at cellular respiration in the second year. This is my summary of it. Briefly, we get energy from food to regenerate the molecule adenosine triphosphate from adenosine diphosphate. You'll notice that all of the macromolecules, carbohydrates, proteins, lipids, ethanol, and ethanol, which is not a macromolecule obviously, are converted to the molecule acetyl coenzyme A. Before this happens, a number of processes need to talk about, which we'll have a look at in a second in some cases. Okay, roles of the gastrointestinal tract organs in general. We'll discuss the specific roles of the organs a bit later on, but we can summarise the general function of the gut into these 4 headings. And we'll continue to discuss this as we move through the lecture series of the various parts of the digestive system. So motility. Motility is a term used to describe the contraction of muscles that mix and propel contents down the gastrointestinal tract. Chewing and swallowing is generally a voluntary process, but after that the process is largely under reflex control. Chewing lubricates the food by mixing with saliva, which also contains the enzyme alpha amylase that starts the digestion of starches. Stomach is a large reservoir that allows the digestion of meal-sized amounts of food, which we'll see in a moment. And while in the stomach, the food is broken into smaller particles, and digestive enzymes start the hydrolysis of proteins. A range of digestive secretions are involved, starting with those from the salivary glands. There are 3 major salivary glands mirrored on each side of the mouth. They are parotoids, the submandibular and the sublingual. You work out from their names where they're located. Saliva moistens food, starts the digestion of food, and also facilitates speech as well. Amylase starts the digestion of starch by partial hydrolysis into smaller molecules such as maltose and maltotriose. We'll briefly mention transcobalamin 1. This is a glycoprotein and is necessary for the transport of vitamin B12 through the acidic environment of the stomach and then into the bloodstream via the lymph. We'll return to this later on as an example of how a micromolecule, in this case a vitamin, is transported through the system. So the stomach, the cardia and the pylorus have sphincters, that is circular bands of muscle that control flow of food into and out of the stomach. The fundus collects digestive gases, the body secretes pepsinogen and hydrochloric acid, and the pylorus is responsible for mucus, gastrin and pepsinogen secretion. As we'll see later, the fundus of the stomach secretes the hormone ghrelin, which is involved in the regulation of appetite. The largest portions of the body, where muscular contractions and enzymes start the digestion of food, and the atrium holds food until it's ready to transit into the small intestine, which more in a little while. We'll note the volume of the stomach swells significantly when food is ingested, a considerable change in size. So gastric secretions. When your body is ready for the digestion of food, such as typically when you're expecting a meal, the stomach secretes 2 molar hydrochloric acid. If you're using this in the lab, you'll certainly be wearing protective gloves. HCl activates the enzyme pepsin, which has a role and also has a role in sterilizing food. The stomach is coated by a thick layer of mucus produced by the gastric mucosa, a layer of cells from the inner surface of the stomach. This gastric mucus consists of glycoproteins, that is to say proteins attached to sugar molecules. It helps lubricate food in order to facilitate movement of the stomach, and also protects the inner line of the stomach from the strong acid conditions. So pepsinogen. Pepsinogen is classified as a zymogen, an inactive protein precursor that is converted either by acid conditions or by a kinase enzyme into the active enzyme pepsin. Pepsin is a protease enzyme, that is, it hydrolyzes peptide bonds in proteins. Pepsinogen, as we can see in the diagram, is secreted by gastric glands, and under the strongly hydrochloric and the strongly acid conditions, it converts the enzyme pepsin, which, as mentioned, hydrolyzes proteins. Again, you'll discuss kinase enzymes in more detail in second year human metabolism, particularly their involvement with ATP. So what happens to proteins in the stomach? The HCl denatures the proteins, causing them to unfold, causing structural changes that expose the peptide bonds, allowing hydrolysis by protease enzymes, as we've summarised in the picture at the bottom. When pepsin has been synthesised, it's said to be an endopeptase enzyme. That is, it hydrolyzes peptide bonds in the interior of proteins. Exopeptidases act on peptide bonds linking the terminal and sometimes penultimate amino acids of a protein, but pepsin is the main one we're concerned about here. Okay, moving on to fat digestion. We're talking about simple lipids here, and refer to the macronutrients lecture for discussion of the structure of simple lipids, if you need a reminder. Lipid digestion starts in the mouth of the salivary lipases and continues in the stomach and small intestines. However, it's quite inefficient in the mouth, as the pH is somewhere from the optimum pH required for the salivary lipase. Gastric lipase works over a broad pH range, 2 to 7, and is quite effective at stomach pH ranges, but the digestion continues in the small intestine later. The process involves the hydrolysis of ester bonds linking fatty acids to the glycerol backbone, and as mentioned, stomach lipases are a bit less effective than those in the small intestine, but do play a significant role in the digestion via hydrolysis of simple lipids. So chyme. The mucosa is full of gastric glands and pits, and there's a prominent layer of smooth muscle, the muscularis mucosa. The contraction of this muscle helps expel the contents of the gastric glands. This layer has 3 layers of muscles, an oblique layer, a middle circular and external longitudinal layer. We don't need to worry too much about the anatomy of that. The contraction of these muscle layers, however, does help to break up the food mechanically by performing vigorous churning motions. Gradually, most solid pieces of food are reduced to a semi-solid material called chyme. The transit time through the stomach depends on the foods ingested. Digestive activity in the stomach promotes the gastroileal reflex, which promotes peristalsis, pushing contents down the rest of the gastrointestinal tract in a controlled manner. This reflex helps ensure the contents of a meal are completely emptied from both the stomach and the small intestine, as we'll discuss later, before the next meal is ingested. But we'll notice the transit times in the list there for various different types of food. About every 20 seconds, the parallel sphincter at the base of the stomach dilates to allow small amounts of chyme to pass in the duodenum. This gradual release allows the acidic chyme to be neutralised by the by the alkaline, that is the basic pancreatic juice. We'll discuss the formation of pancreatic juice later on. Okay, absorption in the stomach. The stomach is not a major organ for nutrient absorption. There is limited absorption of proteins and peptides and no evidence of digested fat being absorbed in the stomach. Simple sugars are readily absorbed, especially if in high concentrations, which obviously has immediate consequence for the blood glucose concentrations. Within about 20 minutes, up to 50% of water is absorbed by the stomach, so facilitating hydration. And ethanol from alcoholic beverages will also be absorbed here. We'll discuss ethanol metabolism in probably the last lecture in this series. So a brief mention of appetite and satiety. You'll discuss this much more in the second year, and you'll discuss, or already have discussed, homeostasis in other modules. Briefly, it is a group of mechanisms that maintain physiological and biochemical equilibrium in the body. Most of the mechanisms that control homeostasis are associated with the action of the endocrine system, with the hypothalamus and the brain acting as a neural control centre. In humans, the major endocrine glands are the thyroid glands and the adrenal glands. Some definitions of key terms. I'm not going to read through them. You can pause the video and have a read of them yourself. Note how they map onto our general physiological condition. Ghrelin. It came as quite a surprise when only about 20 years ago the hormone ghrelin was discovered, and that is produced by the fundus of the stomach rather than thyroid gland or the adrenal glands. Ghrelin increases the motivation to eat. It's mostly produced in the fundus and circulates in the blood to reach appetite controlling structures in the brain, such as the hypothalamus. Significant amounts are also produced in the duodenum. Again, we'll discuss this in more detail in actually a final year module. Ghrelin's name is derived from the phrase growth-releasing hormone, with the Proto-Indo-European root gre, meaning grow, and relin, releasing substance. It's a peptide. There's 28 amino acid residues. It is said to be orexigenic. That is a substance which promotes hunger. Orexigenic is the antonym of anorexia, which is a decreased appetite. Ghrelin is the only known orexigenic hormone. It's released if blood sugar levels are low. And the way it binds to its receptor in the hypothalamus is conserved in primates, rodents, fish, dog, amphibians, birds and us, of course, suggesting its physiological role evolved very early in the history of macrocellular life. As is often the case, most data from clinical studies, in this case of 13 obese subjects before and after diet-induced weight loss. We don't need to worry too much about the details. The point is the influence of ghrelin is clear. After a period of fasting, blood ghrelin levels rise. Following a meal, the levels fall. It is assumed that ghrelin may help the gastrointestinal tract prepare for food consumption, and given regular eating times, anticipates eating. So we can say that ghrelin levels are primarily regulated by food intake. Okay, that's all for this one. Thanks for listening.